Hi, my name is Sarita, lecturer in Botany, Government Degree College for Women, Nalgonda. Today in this video, I am going to talk about development of dicot embryo. In this video, I am going to talk about dicot embryo development. Dicot embryo development. The Vanagra type or crucifera type represents a typical dicotyledonous type of embryo development. Dicot embryo development found in Capsula birsapastaris, which belongs to the crucifera family. Dicot embryo development first time observed by Hanstein in Capsula birsapastaris. He gave a detailed study of this dicot embryo development. The development of the embryo has the following steps. Here, before going to discuss about the dicot embryo development, we have to know about the formation of this diploid zygote. In the embryo sac, three-celled egg apparatus are present at the microphylar end of the embryo sac. This three-celled egg apparatus contains central egg cell and two synergy cells. These two synergy cells present both sides of the egg cell. When pollen tube enters into the embryo sac, this pollen tube releases two male gametes into the embryo sac. One male gamete fuses with egg cell and forms this diploid zygote. Next, coming to the development of embryo, dicot embryo. This diploid zygote takes transverse division and forms two cells. In these two cells, the chalazial side cell is known as the apical cell and microphylar side cell is known as a basal cell. This diploid zygote takes transverse division and produces two cells and forms two cells. They are apical cell and basal cell. Basal cell present at the microphylar end of embryo sac and apical cell present at the chalazial end of the embryo sac. Later on, apical cell takes longitudinal division and basal cell takes transverse division. Basal cell takes transverse division and apical cell takes longitudinal division and forms four cells. Which are arranged in inverted T shape. This is basal cell, this one is apical cell. Apical cell takes longitudinal division and basal cell takes transverse division and forms four cells which are arranged in inverted T shape. This four-celled embryo is called proembryo. This is the proembryo. Later on, apical cell takes another longitudinal division and forms four cells. This four-cell stage is known as a quadrant stage or tetrad stage. The cells of the quadrant stage divides transversely and forms 8-celled octant stage. These 8 cells are arranged in 2 tiers. Each tier contains 4 cells. 2 tiers, each tier contains 4 cells. 4 cells of the upper tier is responsible for giving rise to stem tip and cotyledons, and 4 cells from the lower tier forms hypocotyle. Later on, periclinal division occurs in each of the arctant cell and forms 2 layers, outer layer and inner layer. The cells of the outer layer forms dermatogen. The cells of the dermatogen takes further anticlinal divisions and forms or develops epidermis, and next inner layer cells takes longitudinal and transverse division and forms outer side peribellum region and inner side plerome region. Two cells developed from the basal cell of the 4-celled pro-embryo divide several times and forms a long suspensor cell. This long suspensor cell which contains 6 to 10 cells. This suspensor cell contains 6 to 10 cells. The uppermost of the suspensor, which is present at microphylar end, becomes enlarged and vesicular. This enlarged cell acts as a haustorium. This is the haustorium. Suspensor cell contains 6 to 10 cells. The upper cell is called haustorium. The lowermost cell of the suspensor, which is nearest to the developing embryo, is known as hypophysis. This is the hypophysis. The hypophysis cell takes longitudinal and transverse divisions and forms root cap, epidermis and cortex of the radicle. Due to the further divisions in the embryo proper, the embryo appears to be heart-shaped in the longitudinal section. A continued enlargement of hypocotyle and cotyledons bring about a marked curvature in the cotyledons, and embryo appears as a horseshoe-shaped structure. In mature embryo, the stem tip is terminal and the 2 cotyledons occupy a lateral position. These 2 cotyledons occupy the lateral position. This is about dicot embryo development. This diploid zygote takes transverse division and produces apical cell and basal cell. Basal cell present at the microphylar end of the embryo sac and apical cell present at the lower side, that is chalazal end of the embryo sac. Later on, apical cell takes longitudinal division and basal cell takes transverse division and forms 4-celled proembryo, which these cells are arranged in inverted T shape, forms inverted T shape. Later on, this apical cell again takes another longitudinal division and forms 4-celled quadrant stage or tetrad stage. This quadrant 4 cells again takes transverse division and forms octant stage, it contains 8 cells. These 8 cells are arranged in 2 tiers. Upper 4 tier cells forms stem tip and cotyledons, and lower tier cells forms hypocotyle. This octant cells takes periclinal divisions and forms outer layer and inner layer. Outer layer develops dermatogen. This dermatogen takes anticlinal lesions and forms epidermis, and inner layer takes longitudinal divisions and transverse divisions and forms outer side peribrum region and inner side plerome region. This is about apical cell development. This apical cell development. Next, coming to the basal cell development. Two basal cells of pro-embryo divide several times and form 6 to 10 celled suspensor. 6 to 10 celled suspensor. This suspensor, upper cell of the suspensor is called haustorium and lower cell of the suspensor is called hypophysis. This hypophysis takes longitudinal and transverse division and forms root cap epidermis cortex to the radicle. Next, due to the further divisions in the embryo proper, the embryo appears to be heart-shaped in a longitudinal divisions. A continued enlargement of hypocotyle and cotyledons brings about a marked curvature in the cotyledons, and embryo appears as a harsh shoe-shaped structure in mature embryo. The stem tip is terminal and the two cotyledons occupy a lateral position. These two cotyledons occupy lateral position. This is about development of dicot embryo. Thank you.